Hey now, how's it going? I'm here to talk about the last part of this lecture that has to do with diffusion, and we talked a little bit about water. Now we're going to talk about osmosis. And so let's do that. Again, remember the reason why we're talking about this and making such a big deal about this is that all cells are surrounded by a membrane, and that membrane is semi-permeable. So it only allows some things in, but not everything. And it only allows some things out, and not everything. And that's an important characteristic of life. That's an important aspect of homeostasis, is that the inside of the cell is kept different from the outside of the cell. And that doesn't happen by accident. That requires energy. And consequently, getting stuff into and out of the cell and across that membrane, that's a big deal. And understanding that process helps you understand a lot of things in biology. So that's why we're making such a big deal about this. And we talked about diffusion, and we talked about molecules crossing, that some molecules can cross the membrane, some can't, and we talked about how molecules spread out. And when we talk about crossing the membrane, a molecule that can cross the membrane with little difficulty is water. And it's not, perfectly oh God, that's so terrible. And so we talked about some molecules that are able to diffuse and cross the membrane and get into the cell, and other molecules can't. But one molecule that can get into the cell pretty easily is water. And so water can sort of move across that cell membrane pretty easily. And since it can cross the membrane, and since water is so critical to all kinds of reactions, and it's just so critical to life, we treat it as a special case. And the diffusion of water is called osmosis. And so that's what osmosis is. It's the diffusion of water molecules. And so again, back to our diagram that we looked at earlier. We've got a semi-permeable membrane, and then we've got a molecule that's concentrated on one side. And let's say that this is a big molecule like urea. And because it's big, it can't slip through the channels in the membrane. It can't cross that membrane. And through random molecular motion, that molecule is going to diffuse and spread out on one side of the membrane, but it's not going to cross the membrane. So there's always going to be a higher concentration of urea on one side compared to the other side. And we saw this earlier. And again, that's because, there's all kinds of reasons why that molecule can't cross the membrane. It's too big, or it's got a charge, or whatever. But now we're going to add water to this example. And so the blue molecules that you see here, the smaller molecules, those represent water molecules. And remember, we said water can cross the membrane with little difficulty. And so it can diffuse from one side to the other. Now, when we think about the water molecules, we have to think about them in a little bit of detail, okay? And so if you look at this diagram closely, on the left side of the membrane, on the left side of the diagram, the water molecules are there, but they're associated with urea molecules. They're sort of bound to them with hydrogen bonds. So you remember we talked about hydration shells. And we talked about how things dissolve in water because the water kind of surrounds the other molecule. It surrounds the solute molecule. But consequently, that water is sort of tied up with that solute molecule. It's hydrogen bonds and other things that keep that water associated with that solute molecule. Now, if you look at our diagram, on the right side of our diagram, you don't have any solute. What you have is just water molecules, and those are called free water molecules. And so a free water molecule is a water molecule that's free. It's not tied down. It's not bound to anything. And so it can move freely. And so look again at this diagram. And you can see you've got water molecules on both sides of the membrane. But on the left side, some of the water molecules are associated with the urea molecule. And of course, in real life, there'd be lots of water molecules around each urea molecule. That's what a hydration shell is. But we're just trying to give you just a diagram here to explain something. On the right side, since there's no solute, all those water molecules are free. And so look at how many free water molecules are on the right side, and look at how many free water molecules are on the left side. On the left side, we only have a couple of free water molecules. Now go back to our original definition of diffusion. Diffusion is the random movement of molecules until they spread out and they move down their concentration gradient. Well, water is no different than anything else. It will diffuse down its concentration gradient. And water will move randomly, just like any other molecule. But only free water can cross the membrane. And so although you've got lots of water molecules in our example, only some of them can cross the membrane. And so those free water molecules are able to cross the membrane, but the water molecules bound to urea cannot cross the membrane. And so again, imagine diffusion and imagine water molecules moving around at random, and you can see that the free ones will move at random, and then they might cross the membrane and they might cross back, whereas the ones that are bound to urea will just move around on that one side of the membrane. So only the free water molecules can cross this membrane. Many of the water molecules on the left side of this diagram are bound to urea. They're not free. They can't cross the membrane. They're moving at random with their urea molecule, but just like the urea molecule, they just bounce off the membrane. And so this is just reiterating what we said earlier. Now, if you look at it, you have a higher concentration of free water molecules on the right side compared to the left side, right? And you remember that when we talk about diffusion, things diffuse from high concentration to low concentration. And so in this example, we're looking at the free water molecules, and they're going to diffuse from high concentration to low concentration. They're going to diffuse down their concentration gradient. That's what osmosis is. So there's a higher concentration of free water on one side, and that free water is going to spread out at random, and it's going to be able to cross the membrane. And so you're going to have a net movement of free water molecules across the membrane until the concentrations are equal. It's just like the diffusion we talked about earlier, but now it's a diffusion of water, but not any water, free water. And so why are we making a big deal about this? Well, I already told you. I said, water is important to all kinds of chemical reactions. Water is important to everything. So we like to talk about water, especially. We had a whole lecture, right, just talking about water and its properties. But this is a process that drives many phenomena in biology. And so by understanding osmosis and the movement of water, you can understand lots of things in biology. Okay, so we need some vocabulary here so we can talk about this. If two solutions have the same concentration of free water, they are called isotonic. Iso is same. And so water molecules will be able to move across the membrane, but you're going to have an equal number or an equal concentration of free water molecules in both solutions. A solution that has a higher concentration of free water is called hypotonic. So it has more free water. That means it has less solute. And so you have to be able to think about these two things in your brain, right? You've got water molecules and you've got solute molecules. Whatever is dissolved, urea, sodium, potassium, hemoglobin, whatever. And so you've got the solute molecules and the water molecules. And the water molecules are going to try to interact with that solute molecule and form that hydration sphere, which means that they're not free. So if you have more solute molecules, you have less free water. And so if you've got less free water, a solution with lower concentration of free water is called hypertonic. And hyper, means more, right? You've got more solute in the hypertonic solution, which means you have less free water. Now, these terms only make sense when you're comparing two things, right? You can't say, Diet Coke is hypotonic. Well, that doesn't make any sense, okay? It has to be hypotonic to what, right? Diet Coke is hypotonic to probably is hypotonic to seawater, but it's hypertonic to distilled water. And so these terms only work in a relative sense. And so back to our example, the side on the left side, which has the solute, is hypertonic. It has more solute. Hyper means more. Because it has more solute, it has less free water. So there's less of those free water molecules bouncing around. On the right side, that's hypotonic. It has less solute, which means it has more free water. So you've got a lot more free water. Again, think about those free water molecules moving around. Some of them are going to go to the other side. You're going to have a net movement of free water from the right side to the left side. That's osmosis. All right? So cool. Like I said, water is going to move that way. Water is always going to move to the hypertonic solution. All right? Water is always going to move to where there's more solute. Okay? There's more salt or more whatever's dissolved. So that's an idea you get comfortable with. But if you think about the molecular movement of water molecules, you can reason your way through this if you don't remember which direction the water moves. Okay, so which way will water always move? From high water concentration to low water concentration, from low water concentration to high water concentration, from hypertonic to hypotonic, from hypotonic to hypertonic. Well, there's only two. There's two things are the same, and the other two things are the same, right? So high water concentration to low water concentration. That's talking about free water. That's the same thing as moving from hypotonic to hypertonic. Always, always, always. Water will always move to the hypertonic solution. And so this is important when we talk about cells, because your cells have lots of solutes. Your cells have lots of stuff in them, okay? All those molecules we talked about earlier, carbohydrates, lipids, proteins, nucleic acids, those are all inside your cells. There's a lot more inside your cells than outside your cells. There's also lots of inorganic molecules, right? Things that are on the nutrition label or things that you're familiar with. Sodium, iron, calcium, potassium, so on, so on, so on. There's lots of those kind of molecules. There's lots more inside your cells than outside your cells. And you also do have water inside your cells, but that water is going to be associated with lots of this stuff in your cell. So you don't have a lot of free water, but you do have water. And so based upon all that, is the inside of your cells hypertonic or hypotonic relative to pure water, distilled water? So is it hypertonic, hypotonic, or is this, you can't tell? Well, the answer is hypertonic, right? You got more stuff in your cells. That means you have less free water relative to pure water. Pure water is just nothing but free water. There's nothing else. And so the inside of your cell would be hypertonic. So what would happen if you put one of your cells in a beaker of pure water? So I got a beaker of distilled water. I take a human cell and I stick it in there. We know the human cell is hypertonic relative to the pure water. And so will that cell shrink? Will it swell? Or is it impossible to tell? Well, where's the water going to go? Water always goes to the hypertonic solution, which is inside the cell. So water from the beaker is going to go inside the cell, and that's going to make that cell swell up, right? And so the cell will swell up. This is critically important in a lot of situations. Certainly it's very important like when you're in a hospital, okay? Because if you put your cells, if your cells are in a hypotonic solution, you know, they're going to swell. That water is going to go into your cells and swell because of all the stuff that's dissolved in your cells. But if your cells swell up too much, they'll burst. That's a problem. And so when you get an IV or something, a lot of times you want an isotonic saline solution. Isotonic saline solution means it's saline first is salt, right? So it's a salt solution. So it's water with some salt in it, with something dissolved in it. But it's at the same tonicity, it has the same concentration of free water as your cells. And so if you add that to your, put it in your vein, then the water is going to move equally in both directions and your cells are fine. If you put pure water into your veins, that could be a problem because that water would be hypotonic. And so then that pure water is going to want to go into your cells and your cells are going to want to swell up, and that could potentially be a problem. There's lots of things that can go on, but these are the sort of things that you need to pay attention to. Now that you know these terms, start listening for them a little bit. If you wear contacts, you buy that solution and you got to rinse your contacts and put it in your eye, and most of the time that's an isotonic contact solution. Well, it's got the same amount of free water, same concentration of free water as your cells, and that's a good thing, right? So that way you can put it into your eyes, and your eyes, the water is going to move equally, and the cells in your eyes aren't going to swell up, or they're not going to shrink or anything like that. So this is really important to understanding how the human body works, and all living things work. Okay, why will your cell swell if it's placed into pure water? Because all the molecules will leave? Because water will flow into the cell? Because of the diffusion of molecules? Or it's impossible to tell? Well, again, because water will flow into the cell. Now, think about this. You've got a lot of stuff. There's lots of molecules and lots of inorganic things in your cell. You put it in that pure water. So there's a high concentration of, say, sodium inside your cell and a low concentration outside in this example. Well, remember, we go back to diffusion. Sodium wants to diffuse out, right? Things diffuse down their concentration gradient. So the sodium wants to diffuse out of your cell. That's a basic diffusion that we already talked about, right? That's the first thing that would happen. But remember that the cell membrane is semi-permeable. And so it doesn't, in this example, the cell membrane does not allow sodium to diffuse out. It wants to diffuse out, but it cannot diffuse out. Or you got all these proteins inside your, you got lots of proteins in that cell. A high concentration of protein in the cell, a low concentration outside the cell. Well, the proteins want to diffuse out, but they can't because that membrane won't let them. But the water, the free water, can cross that membrane. And so that free water is going to come in to the cell. That's osmosis. And so here's a figure from your book kind of illustrating this idea. And so you can see on the left you've got a cell, and if you've got a higher concentration of solute outside the cell, so inside the cell is hypotonic, then water is going to leave the cell, and we call that crenation. The cell shrinks. Whereas if you've got a higher concentration of solute inside the cell, then the inside is hypertonic. Water is going to come in and the cell is going to swell up and burst. And so again, that's why it's very important, in medicine and things, like when you're putting stuff in your body, you've got to control the tonicity because it can influence the water flow into your cells. Now, on the right, what they're showing you is a plant cell. And the same thing happens in a plant cell. Plant cells are no different than animal cells, except plant cells have that rigid cell wall around the outside of the cell, okay? And so it's the same thing as with the animal cell. If you've got a higher concentration of solute outside the cell, that means inside the cell is hypotonic, water is going to leave. And that cell is going to shrink and it's going to pull away from that cell wall. Whereas if there's a higher concentration of solute inside the cell, if inside the cell is hypertonic, then water is going to go into the cell and the cell is going to swell. But in this instance, the cell is much less likely to burst because as it swells out, it's going to hit that cell wall. It's going to push against that cell wall, which is going to keep it from bursting. All right? And this is called a turgid cell. Now, here's the analogy, right? Let's say you had a cardboard box and you put a balloon in there. And if you didn't blow up the balloon, you know, that box would be very weak. You couldn't do any, you know, the box, you could push it in. Whereas if you blew up the balloon to where that balloon is inside the box and pushing out, then it gets very hard. Well, that's exactly what happens in plant cells. And that's why when a plant is well watered, you know, it stands up and it can stand up and be rigid. Because every one of those cells has plenty of water, but that the inside of the cell is hypertonic, so that water is getting into the cell, and that cell is pushing against that cell wall, and all those cells are rigid. That makes the whole plant rigid. But if you don't water your plant, what happens? Now there's not enough water. The water doesn't get into the cell because the inside of the cell is now hypotonic, and so the cell is not pushing against the cell wall, and so all those cells they don't have that turgor pressure, and so then what happens when you don't water a plant? It wilts. And so again, by just understanding the movement of water across the membrane, you can understand simple things in biology. Okay, this is also interesting when you think about things like sports drinks, you know, things like Gatorade or whatever, right? Sports drinks are basically water with salt and sugar in them, okay? And so they're water with a lot of stuff dissolved in them. And many of these sports drinks are hypertonic to your cells. And so they've got more stuff dissolved in them than your cells. So what's going to happen? If you drink them, what's going to happen? Where's the water going to flow? If the sports drink outside the cell is hypertonic, the inside of the cell is hypotonic. Water is going to actually leave your cells, potentially, right? And so, I guess I just answered my own question here. What might happen if you drink sports drinks? The sugars and salts will move into your cells will burst, water will leave your cells, which dehydrates you, or you'll dominate the competition, become more attractive, and everyone will love you. Well, I already gave away one of the answers. It's either A or C, or both, okay? So, the sports drinks have sugars and salts in them because you do use some of those during exercise, and so it's replacing them, right? Replacing electrolytes. When I say electrolytes, we're talking about salts. But, like I said, many of these sports drinks are hypertonic to your cells, and if they're hypertonic, then they're going to make your blood hypertonic and they're going to pull water. And so that's actually the opposite of what you want if you're exercising. You don't want water being pulled out of your cells. And so they could potentially have a negative impact. Now, which is it and what happens? It's a much more complicated. And it depends a lot upon how strenuous the exercise is and what kind of shape you're in. There's all kinds of variables here. So you can't say that it's going to be one or the other, or it could be both. My opinion, sports drinks is just Kool-Aid for adults, right? They don't really do much, and there is some data to back that up, that sports drinks, unless you're an Olympic athlete, you know, and you're exercising at a tremendous rate for a long period of time, they don't really replenish anything at that point in time. Really, your best bet is just pure water when you're exercising. But there's lots of variables here. And so again, this is a complicated thing, and you can't just look at one situation and say it explains everything. But there is a low concentration of water in the sports drinks. Well, how do I know this? Because they advertise it. They say, you know, chock full of sugars and salts and electrolytes. If they didn't say that, then it would just be colored water, which it sort of is in a lot of instances too, right? I mean, you know, you have to critically evaluate the marketing behind these things. But if that sports drink has a low concentration of water relative to your cell, that means the water will flow out of your cells. That's not what you want. And so that's a potential response of your body to these sports drinks, but like I said, it's very complicated. There's lots of things going on. But, you know, my understanding and what I've read of the actual studies done on this is that there's just nothing better for you than water when you're exercising. Okay, last thing, this may seem completely off the wall, but what are you supposed to do when a jellyfish stings you? Pee on it? Suck the poison out? Put toothpaste on it? Or soak it in salt water? My guess is most of you probably said pee on it, because that's the classic solution to this problem. But I think all of these are suggested as different things to do when you're stung by a jellyfish. And here's the thing, nothing works and they all work, right? And so it's just like many of the cures for the common cold, or many of the home remedies that we use, is that none of them work and they all work. And the reason is because these things, you give them enough time, they solve themselves, they cure themselves, right? And so no matter what you use, you know, you pee on it, you put toothpaste on it, oh, and it got better. Great. But if you hadn't done that, it would have gotten better on its own. And that's true for a lot of things. And again, that's why we talk about the scientific method, and we try to, you know, there are very specific ways to design experiments to test this sort of thing. But when it comes to jellyfish stings, you know, none of this, nothing, very few things really seem to work, but it's also not something that a ton of research has gone into. So here's the question. Why am I all of a sudden talking about jellyfish? Well, it actually has to do with how they sting you. And the jellyfish are in a group of animals that have these specialized cells called nematocysts. And what these are specialized organelles in the cell, and they have these things called cnidocytes. And the cnidocyte is a structure. Well, the cnidocyte's the cell, and the nematocyst is the structure. It doesn't matter. Anyway, it shoots out a stinging thread, and so that thread sometimes has venom on the end of it, and which causes a reaction. But this whole group, the cnidarians, all have these specialized cells, and it's jellyfish and hydra and lots of other different kinds of organisms. And how this stinger works is all based on osmosis. And so what they're showing you is a hydra here, and it's got these tentacles. And so you can see the similarities to a jellyfish, which is a medusa form that also has tentacles. And if you zoom in on that tentacle, you can find these cnidocytes, and then the nematocyst is a specialized organ in that specialized cell on the surface. And what they're showing you here is inside this, this nematocyst, you've got this coiled up thread that's packed in there very tightly, and it's got a little barb on there, and a lot of times it has venom. And you know, these are very you know, they're not very mobile organisms. They don't have a lot of defense, and so a lot of times this is their only defense, but most of the time this is how they can capture prey. You know, they use the venom to subdue their prey, but also maybe to protect themselves. But they can also have like sticky things on there, so they can shoot these threads out and grab small prey, and then you know, then digest it, or they can shoot the threads out and use them to hold themselves to to the substrate or whatever. So there's lots of different types of these threads they can use, but the ones that sting have a little bit of venom on there. And so you've got this thread coiled up inside there, and then you've got a little cap with a trigger on it. And if you brush that trigger, that causes that string to shoot out, and whatever brushed the trigger is going to shoot into there. And if that's you, that's how you get stung by a jellyfish. Well, how does it shoot out? They don't really have like muscles or anything. So the way this works, the way this uses osmosis to make these threads shoot out and sting you, is that the inside of this nematocyst has extraordinarily high concentration of calcium ions. And so the hydra, the jellyfish, whatever, they expend the energy, they pump calcium. You got a really high concentration of calcium inside that nematocyst, right? And then you've got this cap, and it's got this trigger attached to it. And if that trigger gets pushed, you know, if something brushes against it, like if you brush against a jellyfish, or if their prey or a predator brushes against the organism, it's going to hit that trigger. And that trigger is going to kind of burst open what holds this calcium. And so that calcium is released into the cytoplasm of this cell. And so now the cell all of a sudden has a very high concentration of calcium. That calcium was contained in a part of the cell that's sealed off from the rest of the cell. But the trigger gets brushed, that bursts open that capsule. All of a sudden, instantly, that cell has a lot more calcium in it. It becomes extremely hypertonic at that point. And where does water flow? When a cell is hypertonic, what happens? Water wants to flow into the cell, and the cell wants to swell, right? And so instantly, you've got this cell that has lots of calcium in it, and this trigger has pushed open the cap on this cell, and so water wants to rush in and can rush in. And so immediately, the cell swells up very big, just like we've been talking about, because the cell becomes extremely hypertonic. Well, when it swells up real fast, that is like, it's like an airbag in a car. It's like rapidly expanding it. And you can generate a tremendous amount of force, and that force is used to shoot that coiled up thread. And so when you brush up against the jellyfish, that releases the calcium, water comes rushing in, the cell rapidly expands like an airbag, and boom, it shoots that little thread with a little bit of venom in it. But of course, if you look at this diagram, all along the tentacles, you got hundreds, thousands of these. And so you get a whole bunch of these at once, and it hurts like heck. And again, it could be a defense mechanism. It also could be what they use to capture their prey. Doesn't matter. It hurts when it happens. And so that's what they're showing you here in this diagram. Is that again, if you kind of go through the steps, you can see how in the last part that cell has swollen up and shot that thread out. And that's a jellyfish sting. And so pouring water on it, peeing on it, it's unlikely to do anything. It might dilute the venom a little bit, but by that point, it's injected underneath the skin, and so it's going to be hard to flush that out. Vinegar might work, and it might be due to the pH, not osmosis. It might have an influence on the pH, and so that might work. But baking soda also might work. And again, it would be due to pH. So you've got vinegar's an acid, baking soda the base. So again, you've got this contradictory information. That's why I say everything works and nothing works. Usually it just goes away on its own. And so let's look at some pictures of people who've been stung by jellyfish, which is pretty radical. And so you can see how that tentacle draped across this person's knee, and every one of those welts is where one of those little stingers stuck itself in. And here's a very bad reaction. That looks awful. And here's some, scarring that has come from a jellyfish sting. So, yeah, these things are not pleasant. And you say, well, I live in Kentucky, I don't have to worry about jellyfish. Not true. They're actually an invasive freshwater jellyfish, a little bitty thing that is found in several lakes, and I think it's been seen in Kentucky Lake occasionally. So if you're ever around somewhere and you see something in the water that looks like a small jellyfish, and you're like, "Oh, I'm crazy. That can't be a jellyfish." Yeah, it might be, but it's invasive. It's not supposed to be here. Okay, last thing I want to talk about is an interesting example, again, related to osmosis. And it has to do with water intoxication. And so basically it's what it sounds like. Drinking too much water and the negative effect it can have on your body. And so in 2007, there was, I think, a radio station that had a Hold Your Wii for a Wii contest. Okay? What am I talking about? Well, if you remember, the Nintendo Wii game system was a thing back in the early 2000s. And so this was a contest to win one of these Nintendo Wiis. And the way you had to do it was you had to drink water at constant intervals and not pee. And whoever lasted the longest, the last person to go pee, won the Wii. Hold your Wii for a Wii. Hooray. And so they had this contest, and the woman who was in the contest, the woman who won it, afterwards complained of a headache, and within 24 hours she was dead. And so what do you think happened? So she drank an incredible amount of water but didn't pee any of it out. And she was suffering from water intoxication. And so what happened, peeing out excess water, that's a very important function of your kidneys. And proper water-salt balance is essential to all aspects of living things. And it's very important for homeostasis. And you remember that's what kicked off this whole discussion, is living things have to maintain a constant internal environment different from their external environment. And the amount of water and salt in your cells is important for that, because it's important for lots of things that your cells need to do. If you take in a tremendous amount of water but don't get rid of it, that can have serious repercussions because you're disrupting lots of things in your physiology. And so not just a contest, but there's a lot of times where just, for a joke, or it's just a bad idea. Now, a lot of times you hear about this with like extreme athletes or people that are drinking tons of water while they're exercising, and if you're not careful with this, it's not a common thing. So don't be afraid to drink water. I just told you water is the best thing to drink while you're exercising, and that's true. We're talking about extreme situations where you're pushing yourself to drink water but not pee any of it out. That's a bad idea. It's called hyponatremia. And so what really happens is you get so much water it dilutes the sodium in your blood and around your cells and things because there's excess water. And so you disrupt that salt water balance, and sodium is part of those electrolytes, those ions that are important to life. And so too much water gets those sodium levels too low, and then lots of life functions can't work. Things like nerves or blood pressure get really thrown off because your sodium levels drop so much. So anyway, don't do that. But you can see how the controlling the balance of waters and salt has a lot to do with diffusion, has a lot to do with osmosis. That's critical to life processes, and that's why we've been talking about it. So, all right, that's the end of this section. And so that's all I have. We're going to do some practice problems to make sure you understand this concept of osmosis and make sure you understand that, isotonic, hypertonic, hypotonic, and the vocabulary and things like that. So, as always, let me know if you got any questions. Have a great day. I'll see you later.